Yardi, and thank you, team, for the invitation. It's really great to be back here. So today, I'm just going to share two little examples in my recent work that's related to deep learning. And so deep learning methods are quite a bit different from biostatistical methods. Perhaps it's more similar to Bayesian methods than the frequentist method. But I'm just hoping that these two examples will sort of help you understand a little more what deep learning is all about, and maybe you'll be interested in doing some research in that area. And let's see. So the two topics, the first one is more related to traditional statistical method. It's a non-supervised dimension reduction problem. And the second method, it's a different type of image that we often deal with, not the matrix data, but image data, so image processing. And so I'll start with a sort of a broad categorization of the different types of deep learning models. So we have the autoencoders, which we'll talk about in the first part of this talk. And then they will have the convolutional neural network models, the CNN, which we'll talk about in the second part of this talk. And then there's the transformer models. Those are the ChatGPTs, AlphaFold. And then the generative adversarial networks is a very interesting type of model, which we're not going to talk about today. And RNN is yet another type of model. So it's very versatile. It's a group of methods that's really, really powerful. So the first project is a collaborative work with Jun, who is at Calico. And so this is about dimensionality reduction. And so what dimensionality reduction, the goal is to create either a low dimensional summary of the data or to extract the most salient features of a data set. And it's unsupervised because there's not an outcome you're predicting. And the canonical example, when people think about dimensionality reduction, is PCA. And so this work was motivated by a data set from the study of HIV. And in particular, this data set is it's a study of the mother-to-child transmission of HIV. So if the mother is HIV positive, then there's a chance that her baby will get HIV during birth. And this doesn't happen anymore, but so this data set was collected many years ago when the antiretroviral therapy wasn't yet effective like today and wasn't widely available. So the main goal of the study is to study the biology, to study what's the risk factor when for the transmission. And so, and it's a biomarker study, and the outcome is the transmission. And what we measure is the antibody that's in the mother, in the pregnant mother, that's against HIV. So HIV protein, and HIV has it's a virus, it's an RNA virus with a protein envelope. And the main type of protein envelope protein, it's called GP120 and GP140. And this is shows a side way of the trimer. So it's 3 units bind together. And this is a side view. And this is a top view. You see this 1 unit, second unit, and third unit. And if we look at this side view here, you see that the protein is complicated, and there are different regions that's more antigenic than other places. So they attract the immune system, human immune system. And so one of them, this is the CD4 binding site. So this area is called the CD4 binding site. And this is, and these two are variable loops in the protein. And if we look at how HIV enters a T cell, so HIV attacks the immune system directly. And so this is the membrane of the T cell. On the membrane, you have different types of protein. And one of them, this is the CD4. And CD4, so this is the GP120. This is the same protein that I showed in the previous slide. It's shown here. It's shown as a violin with 2 sets of wings. And these wings are the variable loops. And this is the CD4 binding site. And in this case, the CD4 binding site on the envelope protein binds to the CD4, and this forms a complex, and that's how HIV enters the T cell. So these are the key areas. And in this data set, we have 7 biomarkers, and we can group them into 3 groups based on what the antibody, so the area that the antibody is binding to the envelope. So if so these biomarkers will measure the abundance of antibodies. So the first group of antibodies, they bind to the V3 loop on the protein. And the second group binds to the CD4 site, CD4 binding site on the protein. And the last one, it's not a binding, it's a neutralization assay. It measures how capable of antibody is good at neutralizing HIV virus. And this slide shows a paired plot. So there are 7 antibodies, and it shows that, so not surprisingly, so if we look at the first 3 here, and within this group, they are highly correlated. And if we look at the second group of 3 here, within the group, they are highly correlated. And this is the last one. It's moderately correlated with the other 6 proteins. And so the question is that, are these binding, are these different measurements, are they separate from each other, independent from each other? Is there just one signal, or are there 2 different signals, or 3? And so we can do, if we do PCA, then we get a linear combination of these markers. But as we have seen before, these, if we look at this lowest plot, it's not clearly not linear. So it's not very efficient. So we want to do a way to nonlinear, combine these markers in a nonlinear way. So, well, let's think about the PCA first. Well, PCA is sort of the central dimensionality reduction method, and it can be interpreted in many different ways. How you interpret it decides how you extend it to the nonlinear way. So, but PCA, this is how PCA works. This is how when people use PCA, people keep adding components. So, and then the y-axis, it shows the percent loss, or percent that's not unexplained. So, for example, here, when we allow one principal component to summarize the data, in this case, we explained away about 63% of the variability. There are 37% left. If this is a 20-dimensional data set, when we get to, well, the 20 is not the end here, but if we have as many components as the dimensionality of the input data, we'll be able to explain all of them. And so one way to interpret PCA method is to draw a straight line through the data cloud. And so the natural way to think about a nonlinear way is to draw a curve through the data cloud. And this is the approach was taken by Ducamp and Stewart. And so they introduced the principal curves to formalize the notion of a curve passing through the middle of the data set. And so they define this more rigorously. So a curve is set to pass through the middle of the data set if every point on the curve is the average of the observations that's projecting onto it. And this is all very good, but what's hard about nonlinear dimensionality reduction is the complexity, like how do you decide the degrees of freedom, if you will. So, and this is one comment on why this is a hard problem. So it says, while principal curves are critical points of the distance function, they are never local minima. And so you can't even use cross-validation to find what you like to have. And so in this talk, in this first half of the talk, I'm not going to attack the problem of finding the complexity. But so because of that complexity, there's just a very diverse number of methods that try to solve this problem from different angles. And so we, just to help us understand these methods, we categorize them into 2 classes. So the first type of methods, they emphasize preserving the proximity between the data points. So in other words, nearby points in the original data set should remain nearby in your representation. So when you do dimensionality reduction, you come up with a representation of the data. And so the nearby points in the input should be still close together in the output. And the second category is to minimize a divergence matrix between the observed and your representation. So the second way, perhaps it's a more natural way to think about it for someone who is not haven't studied this, because here we can think about image compression, right? The loss. We try to minimize the loss with a lower dimensional representation. But the first type of methods are indeed actually very popular. So these are the methods that preserve proximity. So the most, perhaps the most well-known method is the multi-dimensional scaling methods, the MDS methods, and there are many variants. And then all the kernel methods and spectral methods, and they all fall under this category. And the second type of method is to minimize the divergence between the output and input. So what we have seen before, the principal curves, principal surfaces, principal manifolds, they all formalize this notion of this representation going through the middle of the data set to minimize the distance between the input and the output. And then you have the generative topological models, and there are some matrix-based methods. And the last one is from the deep learning. It's bottleneck autoencoders. So as I said before, how you look at PCA decides how you are going to extend the PCA. So this is not a way to look at PCA. You can look at PCA as an autoencoder, as a bottleneck encoder with a single node in the middle. Right? So this is your input here. So each node represents one variable in the input data set. And this is the output. And you want the output has the same dimension. What you're trying to do is to minimize the difference between the input and output. And what makes it work is that you go through this layer with a single variable. And so, and these arrows means a linear combination. So this is your PCA component one. You want the linear combination of the input, and then all the output are identical to this code, and you want to minimize the distance. And that's one way to look at PCA. So if we look at PCA this way, naturally, this is how we can extend it to nonlinear PCA. So we add another layer between the code layer and the input layer. It's called the mapping layer. We add another layer between the code and the output. It's called the demapping layer. And then if we look at so every node is a variable. So this, we add an activation function here, nonlinear activation. So this means that, so this variable will be a linear combination of these guys. We have the combination coefficient, we have the bias term, which is also known as the intercept, and then we apply a hinge function to it. So anything less than 0 will be set to 0, and then everything above 0 will be left as it is, the hinge function. So this creates a way to obtain a nonlinear combination for dimensional reduction, and this was proposed 30 years ago. And now for PCA, we want to have one first component, second component, and then so the same thing we can do for in the deep learning. And so one way to do, suppose we want two components, this is one way to do it. We just expand the bottleneck layer with two nodes, so two variables. And so this is, we call it simultaneous model. We just fit this model, we minimize the loss function here. But that's not the only way. A second way is to, perhaps more natural way, is to do it one at a time. So you do that one component autoencoder, and then there's an output. You try to minimize the output, but you can't do a perfect job. So you take the difference, you take the residual, and the stage 2, you try to fit a component to the residual. So that's how it works. So this way you can get two components, and 3 and 4 if you desire. So we call that a sequential model. So what our idea here, so what we do, what we are introducing here is, if we just have one component, what we want is to have a monotone encoder. So this is one way to control the complexity of the nonlinear combination. So so if we call this variable x, and if we call this layer the vector z, and then if we call this, suppose this a single variable, this output variable g, then so z this is a linear combination of the input variable and then with a nonlinear activation function. So that's z. So, and then g, the output is another linear, nonlinear activation of a linear combination of the demapping variables. And so by monotonicity, what we mean is that g as a function of x. So this layer is g, this layer is x. G as a function of x, we want it to be nonlinear. Then there are two, and there are two ways we can do this. The first way is the constrained approach, and the second way is the penalization approach. And in the constrained approach, we can analyze the function form, and then it becomes clear that if we force the combination coefficients to be non-negative, we can achieve that. And the second way is a more gentle method to encourage the positivity. We just put a penalty here, non-linear on the negative part of the gradient, and this can be implemented in the code. So that's the difference. If we have a single component, we introduce a monotonicity to the model. And what about the two components? If we want a second component, so our idea is that we're going to do it. So we call it forward stepwise, but the name doesn't mean anything. But what we're doing here is that, okay, if we want a second component, we put a second component in the code layer. But how this picture differs from the previous simultaneous model is that there's no connection between this variable and the encoder part of the model. So what this means is that this kind of scheme it looks like both has some features of the simultaneous model and has some feature of the sequential model. So like the simultaneous model, the input, so we do it stage by stage, but it's like the simultaneous method. The input of every stage is always the original data. So in a sequential method, the input of the stage is the residual from the previous stage. Here, the input is always the input data, the original data. And the model it used in every stage, or we have the same number of the codes as the stage it's in. But it's also like the sequential method, meaning that the estimation of the component is done one at a time. So this component has already been estimated in the previous stage. We're not re-estimating it, it's fixed. So by including the nonlinear components estimated from the previous stages, the proposed method, it provides sort of a consistent measure of the reconstruction error through every stage. So this will become clear in the example. And another thing we want to do here is that in PCA, we want orthogonal, we want uncorrelated components, right? And so here we want to have the same thing, so we can define what orthogonality mean. And so one way to define it is one way to promote that feature discrimination is to add another penalty term that's proportional to the L1 norm of the covariance between the current stage nonlinear component and the nonlinear components estimated in the previous stages. And then with both, so now we have in our last function, we have two penalty terms, we have two tuning parameters, but what the selecting the tuning parameter in this problem is quite easy. It's very robust. It's not critical to the problem, to the performance of the problem. And so I'm going to show a Monte Carlo study now. And so this study, I'm calling it SIM9 because there's 9 observed variable, but it only has code 1, code 2, code 3. So 3 signals, 9 observations of those 3. And this shows the pairs plot showing that this is group 1, this is group 2, this is group 3. And then this slide shows that when we do the monotonicity constraint, we have 2 methods. We have the constraint method, we have penalization. So this is a side-by-side comparison of the 2. And so the x-axis is the number of the components we allow in a model. So 1, 2, 3, 4, 5, up to 9. And then there are 100 curves here. That's from 100 different Monte Carlo replicates. And the y-axis is the mean squared reconstruction error. And because we standardize the input, so when we have 0 component, this MSE is 1. When we have 9 components, it's 0. So so the range is from 0 to 1. And so it's very easy to interpret. So this, for example, this means that with 1 component, we explain 60% of the data, and the 40% we cannot explain. So if we look at 3 components, we know the truth is 3 components. So this is 3, and this 3, and this box plots, it's from 100 replicates of the constrained method and then 100 box replicates of the penalized method. So we see that the losses, the MSE is higher for the constrained method. It's lower for the penalized method. So the gentle way to penalize, to promote the monotonicity actually works better because you achieve better fit to the data, better fit with the same number of the components. You can explain more of the data. So from now on, we're gonna just use the penalization method to promote the monotonicity. And so this is the analysis of the motivating data set. And we have 7 variables. So at 7, every method should be able to explain 100% of the variability. And this gray line is the PCA. So with 1 component, it's pretty, there is a lot of correlation. With 1 component, we can explain 75% of the data for PCA. With the proposed method, we can cut the unexplained part from 25% to 15%. So that's what nonlinearity gives you. And then we compare 2 different methods. There's a sequential monotone method, and then there's the one that we favor the forward stepwise method. And so with 1 component, there's no difference between the dashed line and the solid line. But as we add in component, you see the forward stepwise architecture actually works better than a sequential method. It's more efficient. And so with 3 components, the forward stepwise monotone autoencoder explains 95% of the variability. So this is another way to examine what are we extracting in that first nonlinear combination. So the y-axis in both panels are the PCA first component. The x-axis on the left here is the first component from the nonlinear combination. So you see a slight nonlinearity there, right? It's not very significant, but it reduces the unexplained from 25% to 15%. And in this panel here, it's a different method. It's the kernel PCA method. So if the kernel PCA method, when you do a scatter plot with the PCA first component, you see this kind of relationship. And so here you see a monotone relationship, and that's because we enforce the monotonicity. And this makes the kernel PCA first component a little harder to interpret, if you will. And another thing we can do with the dimensionality reduction is to cluster the features. So the x-axis here is the input variables, the y-axis is the 7 components we can extract from there, and we can do a correlation. Looking at a correlation between each of the input and each of the latent variable, if you will, you see that the first latent variable, the first linear component, is highly correlated with everything. So one of our original questions is that how many signals are there? And this seems to suggest there's only 1. That's the C1. And so this data set is embedded in a study of biomarker study between the risk of transmission and the biomarker. So we can look at how the correlation, if whether we can do regression to look at if any of the input marker is correlated with the risk, and we can also do that for the component. So if we look at just take the top 4 component, nonlinear component, and do regression with the transmission outcome, we see that only the first component is correlated, is correlated with the transmission risk, meaning that if the pregnant mother has a higher value in the C1, the risk of the transmission is reduced. The other 3 here, they are not correlated. They are not explaining that. And so, and just to give you another example for comparison. So this is a different data set, and this is from a randomized efficacy. It's a phase 3 trial to look at HIV vaccine. The vaccine itself is not effective, but we study the vaccine elicited antibody to see in the vaccine arm if the antibody level has any relationship with the risk of HIV infection in the vaccine arm. So in this case, data set, we have 7 markers, right? So I'm not going to go into details about what these 7 markers means, but then we do the nonlinear combination. So we are just looking at first 4. And so this is looking at how the features cluster, and we see that the first component is clustered. So these 1, 2, 3, 4, 5, these 7 is clustered with the first component, and this variable input feature is clustered with the second component, and this variable is clustered with the third component. So in this case, there are 3 different components, it appears like. And if we look at the risk, the association between the risk of HIV infection and the biomarker, we see that there are 2 signals here. The first component and the third component, they are affecting the risk of infection, but not the second component. So in summary, the 4-word stepwise monotone autoencoder, it's a nonlinear dimensionality reduction that seeks to minimize the reconstruction error based on a parsimonious representation. It imposes monotonicity constraints to help extract more interpretable components. So if you think about the monotonicity, it means that every latent variable, every observed variable is a positive combination of some of the latent variables. So you can have wiggles in there. So that kind of constraint puts a cap on the type of degrees of freedom you can have, and which stabilize things and make things more interpretable. And so, and the proposed method uses a sort of a new, perhaps more natural network structure to extract multiple components, by including components estimated in previous stage when we're estimating a new component. And then, so with this, with this procedure, in the end, we get a MSE plot. So the mean squared error versus the number of the components, the y-axis between 0 and 1, and has a very clear interpretation. That's sort of like PCA. You can use that to choose a number, to choose the number of the components if we are but without any theoretical justification. If you're looking for elbow point, for example. And then the components estimated, and just like any nonlinear dimensionality reduction, it can be used to cluster the features. It can be used for downstream analyses. And these are a few main references. So this is the first part of the talk. Do you have any questions about this part? Yes.
Yeah, it's really interesting. Thank you so much. I have a question about sort of the fourth point in the final point in your summary idea, where you're talking about using the components to for downstream analysis, for example, for predicting transmission instead of your original variables. And I was wondering if you could just tell us a bit more about the motivation for doing that, as opposed to conducting, for example, a supervised deep learning problem where you're using your 7 input variables to predict transmission. Like, why would you use, what's the motivation for using one of these components of some fitted model to for prediction as opposed to the original set?
Right. So good question. So if you do it, you can do supervised learning. The goal is to learn the best way to use all the information you have in order to predict the outcome. So that's one objective. The objective here is slightly different. The objective here is to, because because there's certain structure to the data set here. So for example, the V3, as I introduced in the beginning, so you can group it by where the antibody is attacking at the surface of the protein. But within this class, you have these different V3M, V3B, and V3. The differences there is because of the diversity of the HIV variant, circulating variant, and that's the thing that makes HIV very difficult to work to make a vaccine. So there's that diversity. So what we are trying to so to me, the question here is that the antibody that's generating by the vaccine, it's not providing any protection. So the vaccine efficacy is not working, but there is a correlation between the risk and the antibody level. So here, my curiosity is to say, what matters here? Does it matter? Do we have, if does it matter, like, there are two different variants, for example. Those are, if binding to one variant and binding to a second variant, are those both effective, or are they, it's the similarity that matters? So that's the number of the latent variable, independent latent variables that's correlated with the outcome. That's the thing that we can sort of try to answer from this. Yeah.
Thank you. I had a question about your second to last bullet discussion point about selecting the number of components. Could you go back a couple slides to that plot of the MSC? Yeah, because you made a point here about how much better the MSC is at one component, but you presumably would not select the one component model. So if you apply some heuristic selection of number of components to these three lines, would you select a different number of components under the different? Like, are the results really that different? Is really my question.
Yeah. So, so if we say 5%, so if we say, okay, we are happy with 5%, that's not unexplained. That's not explained. So we draw a horizontal line here. So, well, so we get 3 components for the nonlinear methods, and we get 4 components, say, for PCA method. So the difference is not huge, right? But those components, perhaps, there is a sort of the PCA, 4 components. So those 4 components work together to explain those 95% differences. Yeah, I understand what your question. I'm not sure I have a good answer. Yeah.
No, it's helpful.
Yeah.
Is this cross-validated or is this on the training set? I guess on training set it goes to 0.
So the so it's been a while. So the autoencoder, there's not an overfitting problem because it's the bottleneck layer limits sort of what you can do, the complexity of the model. Like if you it's like PCA, like you fit the model to the data set. If you just allow 1 degree of freedom, you can't overfit. So the same thing with the bottleneck, I think. With the bottleneck, you can't overfit. So it is on the same, there's no training testing split here. It's self-trained. It's sort of, it's not supervised, it's not unsupervised, it's self-supervised. So we fit the model to the data set, and then we look at the, compare the output and the input, and that's what's on the y-axis.
So PCA, you could train a training set and then have the validation set to explain less variability or a fixed number of components.
Right, the same. Okay, yeah, okay, yeah. Yeah, so this is not it. This is the same data set. Sorry, could you speak it one more time?
Can you go over the difference between the 4-step framework and
Yeah. So this is the 4 stepwise. So the difference between this and which one? The simultaneous?
Yeah.
Okay, so in the simultaneous, so if you look at the simultaneous here, so this node is connected with everything in the mapping node. So there's no difference between these two. These two are, there's a million ways these cut, it's totally non-identifiable. But even that sort of, there's no, so in PCA, you have component 1, component 2, right? Component 1 explains a lot, component 2 explains a little. Here, the 2 components are symmetrical. So that's one motivation for doing it. So in here, this is already done. This node, it becomes the data. It becomes, so when there's nothing in here, that's in the data set. So this is the data. In stage 2 fitting, this is the data. So no, the monotonicity is the penalty, or if you will, you can constrain these combinations. So yeah, so the monotonicity, you can put that even if you just have a single node, you can enforce that. Yeah. Okay. So how much time do we have? 20? Okay. So the second topic is totally.
Could you actually
Yeah.
go back to slice this one?
Yeah.
How are we supposed to interpret a significant p-value in this context? Are we modeling one of the biomarkers and saying the learning principal components are significant in predicting the biomarker?
Oh, so
Or
yeah.
What does this model fit to?
Yeah.
Are these p-values associated with coefficients in the model?
Yeah. So this is just a logistic regression with a 2-phase sampling, and so the outcome is the HIV infection, and the variables are some of the demographics and plus the biomarker. And so this is the odds ratio. That's the p-value. That's the confidence interval for the coefficient for the odds ratio.
The C1, C2, C3, those are not learned features from the autoencoder.
Oh, it is, sorry. So C means, yeah, C means code. So this is the first nonlinear component. Yeah, it's a feature we extracted from the data. Yeah. So the second topic, it's the data is image data, and this is a joint work with Sang Wu, who is now at Miami, U of Miami, and with Dr. Jia, she's an immunologist. Kay is a technician in her lab. So what the cell segmentation is supposed to do is that, given a picture like this is a fluorescence image of PBMC samples, you're supposed to segment out, just cut out where the cells are. So for reasons like you can study the number of the cells, or when you have this here, it's more interesting. Perhaps this is a HSV, so herpes simplex virus, and it's a virus, human virus, and then so if someone gets infected, you get lesions on the skin, and if we take the lesion skin sample and look at under the microscope to see how the immune system is combating the virus, you can stain with different markers. So CD3, CD4, CD8, and this is one of the, I think it's CD3 marker. You can see the immune cells in action, and you can study how the immune system works. And so here it's a lot harder to segment those cells with the methods that you just get from the GitHub. And so, so this is how we measure the performance. So it's called the average precision AP, and it's the true positive divided by true positive plus the 2 types of negative, the false negative and false positives. And when we compare a predicted cell and a true cell, and the true cell come from human annotation. So we have to say, okay, we have to choose the threshold to say how much overlap means a match. So often we use just 50%. So 50% IOU means a match. And then based on the match, we can count true positive. We can get AP. So AP is between 0 and 1, and that's our metric. And so I don't have time to talk a lot about sort of the methods behind these, but it's interesting. I think this is really interesting. And so to me, there are 3 main approaches to do the cell segmentation. The first one comes from the computer vision. In computer vision, people segment anything, right? And then so you can just use the same method to segment a cell. Now, on the other hand, in the cell segmentation field, has a longer history than deep learning. So before the deep learning, people are doing these. And so there was a traditional method research that's based on distance-based methods, and there's the gradient-based methods. And those, when deep learning comes along, those got married to the deep learning. And these method is one of these methods to me, it seems like has the best performance. So just quickly, the computer vision, at the beginning, people are excited about being able to tell the difference between cats and dogs. That's the classification. And then later on, people get into segmentation. Yeah, there are different type of segmentation. And what we're interested in is called the instance segmentation. And so a lot of the early methods, a main branch of work on image is to use CNN. The other one is to use the transformer, but we are focused on CNN here. It's convolutional neural networks. So we talk about the different types of computer vision tasks. There's the object detection, instance segmentation. It's all very confusing at first. So, but the main difference, the object detection is to just put a box. Like if you have a cat, put a bounding box around the cat and you're done. You don't need to worry, but cut out the exact boundary. And then instance segmentation just says that, oh, you need to tell me which pixel is the cat, which pixel is not a cat. But either way, in computer vision, when you do instance segmentation, you first make inference about the box. In the native self-segmentation methods, no boxes. So there was a 10-year window when people are doing crazy work on this. So you have RCNN, a year later you have fast RCNN, a year later you get faster RCNN. And it's not very creative, but it's easy to understand. So I'm skipping over these. And then finally, you have the mask RCN, which does the instance segmentation. And so on the other hand, we have these native self-segmentation methods. So they don't do boxes. They go straight to instance segmentation. And so when the deep learning comes along, they use the convolutional models to predict pixel-wise auxiliary variables that can be used to make inference about cell masks. And so one thing that every method predicts is that whether each pixel is inside or outside the cell. And then different methods differ in how do they augment that space. So cell pose augmented with gradient flow, and the deep cell, they augmented with distance to the cell center. So with either one of those, you can guess where the cells are. So I'm going to talk in a little more detail about the line of work that leads to cell pose. So in 1995, people started doing image intensity analysis based on histogram. So if you have well separated, if you have bright cell, dark background, well separated, you can it's very easy to do cell segmentation, right? So the histogram method works, but people get more ambitious. So people figure out, well, if we look at the gradient, so each pixel has an intensity, and you can derive the gradient, and if you track the gradient, it will lead you can use a watershed method to find the basin, and then each basin corresponds to one cell, and that works pretty well until people get more ambitious. Now here you have cells that are very close together in the crowded region, and the original method doesn't work very well at the boundary. And so some really smart physicists come up with an elastic deformable model. So they transform the gradient vector into gradient flow. So this gradient flow, it's more robust than the gradient vector field. That's only based on the pixel intensity. And it's more likely that if you follow the gradient, it will point to a sink. So that's the sort of the state of the art before deep learning comes along. When deep learning comes along, And people took the idea. They say, okay, cell post take the idea. So they're going to, given a picture, they're going to predict a gradient flow from the picture based on mix of pixel intensities. But to train a model to do that, you need to have a training data. So in the training data, you need to have the gradient flow. So these methods, they don't directly predict cells. They predict inside, for each pixel, they predict three things. One is it inside a cell or not? Can be any cell, inside a cell. And then it predicts a gradient flow, X direction and Y direction. And then you need, when in training, you need to give it a training data. How do you get the training data? The training data it's not based on the image. It's based on the cell map. Like a human comes and based on the image, they draw the mass, cell mass. And the shape of the cell mass can be used in this, this simulated diffusion step. So this is what the illustration. So this is the shape of the cell. And then this simulated diffusion, it's just a mathematical process. You can define, and then you come up with two variables for each pixel. So no intensity is even used. Intensity is the input. It's used to train this. And so I'm not sure if I'm confusing you or not. It's probably, but so the actual network, it's very commonplace nowadays. It's a UNet because unlike classification, you're not predicting a single attribute from an image. You are predicting a set of attributes for each pixel. So you need to go down in spatial dimension and then you have to climb back up. So it's a UNet. And then, so there's more details about the UNet. And this is about a different method, the deep distance. So that gradient is not the only approach you can do this. You can try to model, so this is the image. So this is the image, cell center cell. You can try to, if you can estimate, predict for each pixel how close it is to a cell distance, then you can there's a lot of information there. You can infer which pixel belongs to which cell. And so this is a totally different way to do that. So our motivational data set, like I have shown you before, like that. And so we have, so first of all, we need to annotate the data. So first of all, we tried what's available and they perform better than we expected. They perform at 50% of the accuracy. So if you look at the AP, you get it done about half of the time. And after the val, it's not good enough to use actually. So we need to figure out a way, how do we get to a place where it can actually be useful? So we decided we want to train the data. So we want to fine tune the data. And so we need to have the fine tune the model. We need to have the training data. So the model this page shows what training data we have. So in the end, we collected about 12,000 masks. So human made drawn mask. So this is drawn by a technician in the lab. And so this is kind of a bottleneck or the training because this is not very expensive. But if you think about medical imaging, you need to annotate the CT. The people who can do that, their hourly rate is very, very high. So, but luckily here, any of us can do this without too much training. So we have 10,000, we split into 12,000, we split it into 10,000 training instances and 2,000 testing instances. And this is to say that people often do the segmentation based on nuclei and then expand the nuclear segment mask. It's not working here because in skin, the immune infiltrate, they don't have good nuclei for you to work with. And so this shows there are 18 pre-trained models we have used. First to look at the performance and then these are the summary of the performance. So this by image, 7 images. And if we average over them, we get mean AP. So this is the only column we need to focus on. And as I mentioned before, about the best model is the cell post model. Cell post has a model it's called Cyto. So it's trained from 6 types of very diverse images. Some are microscopy images, some are just apples and oranges. They want to get a model where it's not overfit to a particular type of model, and it's working very well. So it's the best model when we just use it. And then they also train the Cyto model. They start with the Cyto model. And so the cyto model was trained with about 50,000 instances, and they're a lot bigger. There are some data sets like TissueNet, live cell, they're a lot bigger. So they took the CytoNet and further trained with these data set, come up with additional model. They have the zoo. And these models don't work well. They're overfitted to those big data sets. They don't work well for our data set. So, and then deep cell, it disappoints. And then this is the mascot CN. This is from the computer vision. It performs decently. Right? It is something that's designed to work with everything, and it works not bad for cells. And then so in some of these models, they are trainable. Different groups, they share things at the different levels. And for some approaches, we have to come up with a model that's trainable. We have to train a model that's trainable, and then we have to fine tune with our own data. But in the end, this is what the training brings us. So at 0 means that that's the model is already there, and this is the process of fine tuning. So we have a total of 10,000 training instances spread into 7 images. And the group of green are some of the cell post models, different cell post models. And the gold ones are the deep cell models, and the red one is the mascot CN models. So at the end of the training, so train 7 is at the end of the training, the cyto model. If we start with cyto model, after 10,000 training instances, we approach 70% accuracy, starting from about 52. So, but when the lab people look at the image, they are very happy. So 70%, they are happy with 70%. They're not happy with 50%. And if you compare with the other methods, and the models, if we start with TissueNet, this is the model that's trained to analyze TissueNet. On our data, it's starting with 0.1%, but after training, it gets up to almost 70% as well. And then with the mascot CN, it's about 60%. So cell post is the best. So this just shows sort of what kind of the knowledge of the model, if you will. So the right axis is the metric, right? The MAP. The x-axis is number of the training instances. So on the very left, the first one is that if we take the cell post model without any data, not trained with cyto database, just train it with our own data, a small data set, 10,000, we get 60% performance. Now, if we take the cyto database, that's the 50,000 instance trained model, we get 50%. And then from 50%, if we fine tune with our data, 10,000 data points, we get 70%. So this is the best. So the other thing, so if the cyto is already trained to fit a different model, it performs very poor on our data set. But it doesn't matter. If we fine tune with the data, we can mold it to learn about our features. It doesn't lose the generalist model. So it comes back to about 70%. So I thought that was very good, nice feature about the cell post model. And there are deep learning models. They have different tuning parameters we can optimize. And the only thing that's interesting that came out of this is that the data augmentation, the standard set of data augmentation does rotation, flipping, scaling. It turns out, in our experience, random rotation hurt things. And some reviewers some people didn't thought that was strange, but actually, when we look at the medical imaging literature, this is very common. They never do random rotation. And so this is just to get an intuitive idea about what's working, what's not working. So it's maybe hard to see. So the first panel is the truth, is the ground truth. And the second panel is the cyto, and it's the gold masks true positive. And this is the trained, fine-tuned cyto. I don't know if you can see, there are more gold masks. And this is deep cell. It has a lot of the green. I think it's the false positive. And then this is the mascot CN, and it just doesn't have as many gold masks. And if we look one region in detail, we see that, so this is cyto before training. And in ground truth, there are 2 cells here. After training, we were able to, so here, after training, we're able to see there are 2 cells actually here. So when the cells are clustered together, it was hard. it's hard. But with fine tuning, we can overcome that. And then so it can do a better job recognizing 2 cells versus 1 cell. And but it was surprising to us at first, why is it only 70% accuracy? Shouldn't we get 100%? And then we tried different things, and then we realized that the signal to noise ratio in this data is just that great. So if we ask a person to do it twice, what's the correspondence? What's the performance for the second person? If we treat the first person as the ground truth, it's about 70%. So, and this just to show that for this image, a person do it twice, they get different answers. I don't have a summary slide for this page for this. And so that's the end of my talk. Thank you very much.
Questions? Yep, sorry. Maybe shut up.
Sorry, what's your, let me go back to the slide.
Yeah, it's true positive, false negative, false positive, right?
Ah.
So you don't get a true negative. So the information
Oh, yeah. So it's weird to think about true negative here. And so if we use the human annotation as the ground truth, and then so we have a set of masks, and then we compare with the set of predicted mask. So I'm not seeing the true negative category here in this case.
But when you're comparing all these different methods, do they have the same true negative correction?
Comparing.
So you were comparing like different, like the side of the different approaches.
Mm-hmm.
Do all of them have the same true negatives?
So how do you define true negative here? Yeah.
But you define false negative, right?
Huh?
So you define false negative.
False negative, yeah. So false negative means that when human draw a mask, but the model is not predicting a mask. Yeah.
So that's not the possibility of a true negative.
I may be missing it. I'm not seeing it. Yeah.
Any other questions? Not then let's thank Youyi again.
Thank you.
Very much enjoyed the first part. Actually, I've been thinking. First of all, it's only recently I realized that you can think of these other encoders as principal curves methods. Because I was thinking of implementing principal curves directly using sort of neural network or approximated Boolean trajectory, but then I was like, how do I define the projection of the point on the curve, right? It's complicated. So then I was like, oh, but that's what autoencoders do. They usually use the, you can think of the encoder as doing exactly that, right? Taking your points and calculating where the projection along the curve is, and then the other, right?
Exactly.
Yeah, it's only recently that I came to this. Great talk, yeah. And can you think of incorporating also things like smoothness of the principal curve?
Smoothness?
Yeah, when people use pings, they have some secondary derivative smoothness so that your base model could become linear and you could go back to getting PCA as a simplest model and then you allow for more complexity.
Yeah. So that's something the loss function can be. Like, you mean
exactly, you can do it
yeah.
in loss function.
Yeah,
Right?
yeah.
Thanks.
Thank you.